Hello, everyone, and welcome to today's live broadcast. The Pandora's Box of Molecular Diagnostics, Genomic Analysis of Uterine Lavage Fluid Detects Early Endometrial Cancers, presented by John A. Martignett, MD, PhD. We are excited to bring you this educational web seminar presented by Wrapup, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. I'm Julie Simmeroth of Wrapup, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please note that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. Now, I'd like to introduce Dr. John Martignett. Dr. Martignett, an expert in human genetics and genomic sciences at the Icahn School of Medicine at Mount Sinai, as the network director of the Laboratory for Translational Research at the WCHN Biomedical Research Institute. Under this collaboration, Dr. Martignett's well-established bedside-to-bench cancer research methodology will be extended to patients at Danbury, Norwalk, and New Milford hospitals, leading to more personalized and successful therapeutic strategies. He will work closely with the oncology physicians and research scientists across WCHN to build their nationally recognized efforts. Please join me in welcoming Dr. John Martignett. I will now turn the presentation over to him.
Julie, thank you very much for the introduction, and thank you again to you and to Wrapup for the opportunity to speak to everyone. So the talk that I want to give today is really based upon the most recent research that we've done. And I've entitled it probably with the world's longest title, and hopefully people will forgive me for that. But I do hope something is to be gained from the information presented. And really, we're calling this the Pandora's Box of Molecular Diagnostics, and it describes our efforts to develop a screening and diagnostic test in endometrial cancer, and really our success, but really is the second part of the talk, describes really the promises and the perils of advanced molecular diagnostics as they do go hand in hand. So I do have a disclosure to make. Swift Biosciences provided the technical support in part for these studies. So, as we think about genetics and genomics, really the 21st century medicine question is, can we provide personalized or precision cancer care? So, in this cartoon that I have, here's a number of women that are all represented. They all look very similar. When you look at the type of clothing that they're wearing, but you do see that there is one person different. And clearly medicine is based upon understanding large numbers, of individuals and arriving at a diagnosis, arriving at a treatment plan. But there are times when we really need to think about the individual, the one person. And how do we go about doing that? Well, clearly as the entire focus of personalized medicine, liquid biopsies, genetics, genomics, it's really trying to think, at least in the cancer field, is trying to think about a patient's specific mutations and using that information for an endpoint, maybe therapeutics, maybe monitoring, maybe a number of different things. But the idea is to treat each patient's tumor as an individual collection or data point, so we can understand the mutations that are there and potential treatments, outcomes, differences in that particular patient's tumor. So with that being said, the lab has been quite successful, and we've been quite fortunate to work both with outstanding physicians, in particular Peter Catino, who's the Director of Gynoc Services at Mount Sinai, and now Vagan Edikian, who's the Director of Gynoc here at Danbury and WCHN, in using ctDNA, so a different part of liquid biopsy than I'll be talking about today, but thinking about the use of ctDNA as a biomarker and also as a surveillance tool. And here's some of the work that we've presented in the past. It's not what I want to speak about today, but it does provide the framework for what we want to talk about. And so the clinical problem we're talking about is endometrial cancer. Now, endometrial cancer is the most common gynecologic malignancy and the fourth most common malignancy among women in the U.S. In 2017, this year, there'll be approximately 55,000 new cases diagnosed with about 10,000 deaths. Strikingly, the incidence is increasing. It's been estimated that by the year 2030, endometrial cancer will actually be the third leading cancer among U.S. women. Now, the symptoms are really, again, as with many cancers, are a little nondescript, but there is one particularly interesting one where the symptom is non-menstrual bleeding or discharge. Heavy bleeding, dysuria, pain during intercourse, pain or mass in the pelvic area, weight loss, back pain. But again, this postmenopausal bleeding is probably the number one reason for a woman to visit a gynecologist, and about 10% of those women will turn out to have endometrial cancer. Obviously, 90% won't, so it becomes a diagnostic dilemma. Now, the diagnosis is really then based upon pelvic examination, endometrial biopsy, transvaginal ultrasound, but really the gold standard here is hysteroscopy performed in the operating room, followed by dilation and curettage. In other words, you're taking a piece of that tissue and sending it to the pathologist for their evaluation. There are no clinical useful molecular markers for diagnostics or screening. Now, one may ask, well, why OR-based, operating room-based hysteroscopy? Well, in-office procedures do have failure rates, depending on the age of the patient, what the other diagnostics or diagnosis may be. Upwards of 15% of cases may actually be missed. Now, the issues with the operating room, while it is the gold standard, clearly there's cost, there's the risk of general anesthesia, there's pain, there's tissue sampling issues, and there's really, at the end of the day, the test, it's a subjective interpretive test. Now, clearly at Mount Sinai here at WCHN, we're quite fortunate to have outstanding pathologists with experience. Now, again, depending on where you are in the world, you may not have that luxury. And even within such really, ivory towers, there is room for misdiagnosis. And that misdiagnosis is not necessarily the pathologist's fault, but it really is our understanding of endometrial cancer. So the other thing I want to get across is that the pathologist is not only looking to say yes or no to the presence of endometrial cancer, but it's also the type of endometrial cancer. There are two types, and this was really from a grading system developed about 30 years ago, where there's a less aggressive form, type 1, and it's usually a lower grade type of cancer, and type 2, a more aggressive form, and those are the higher grade cancers. At the end of the day, the classification of type 1 versus type 2 and the diagnosis of endometrial cancer is really, let's see if I can get one of these fancy tools here to work, is really subjective test. It's the pathologist's experience and the availability to have a microscopic piece of tissue to look at and to make the call. So with that in mind, I want you to think about why that becomes important. Again, with 55,000 cases per year in the U.S. and rising, and about 230,000 cases worldwide every year, what's happening is a patient will come into the office for the diagnosis. They may have whatever symptoms may be of the list that I've shown you. In office, the physician must essentially get a little piece of tissue to start the process. That goes to pathology. The pathologist makes a subjective call. when they go to the OR for surgery, for hysteroscopy, for whatever they're in the OR for at that point, that subjective test becomes very important because then it leads the surgeon on the type of surgery they will do, whether they go quite deep to actually get lymph node biopsies or not, depending on whether the call was made for type 1 versus type 2. But it will also have implications for the type of treatment, whether the patient will just get vaginal radiation or may have vaginal radiation plus chemotherapy on top of that. And what we know clinically is even with the best of diagnoses and the best of intentions, there are always outliers. For example, I have here, this is a patient from our hospital system, patient 361, was diagnosed with type 1 endometrial cancer, which should have been the more benign, better outcome cancer. Within the first year, the patient had recurrence of their disease. This is very unusual for a type 1 patient. Unusual because it's not what type 1 should do, but with our surgeons and oncologists, it's not that unusual. And so that's what we're really trying to think about here. Are there ways to do diagnostics, prognostics, where one, we can detect cancer earlier, because the earlier you detect them, the better the patient will do? And can we think of a way of understanding, maybe molecularly, the difference between the less and more aggressive forms of endometrial cancer? Because ultimately our goal is to have patients survive better and have a better quality of life. Now, the TCGA a couple of years ago did a fantastic job by looking at a large collection of endometrial cancers, and they were able to essentially distinguish not just 2 classifications, but really 4 classifications. And you see that in the little cartoon there on this slide. That was important because based on the classification, based on whole exome sequencing, RNA-seq mutation analysis, you could kind of rank patients by their survival expectations, and that's what you see on the lower right-hand side of this slide. Now, that's fantastic. But if you also go back and you look at the data, which we actually had, we're quite fortunate to have Boris Reeve and Dmitry Ikonov, biostatisticians with us at Mount Sinai, who took the same data and said, well, wait a second, what if we just look at the pathologist's call and make the distinction on survival there? And you can see the 2 slides, the Kaplan-Meier slides, are very similar. One could almost even argue that just based on pathology or the tissue characterization, you actually do a little bit better in predicting survival. But even if you do that, it's still not perfect, right? You want to be able to say yes, no, not likely. But what's important here is that the molecular does support the tissue diagnosis. So if we can get better with the molecular, we really should be able to refine something. And so our question was, can one develop a molecular diagnostic to diagnose and improve the accuracy of separation between type 1 and type 2 endometrial cancers? And Boris and his group actually came out with a methodology called molecular signature method. Now that we had the concept of using a panel to screen and to use it for endometrial cancer diagnostics potentially, we came back to our original question. Can we define personalized biomarkers? And the whole concept of the study really was hinging upon our ability to define panels and the concept of how do we get material to potentially screen women for the presence or the earliest presence of endometrial cancer. So if you think about the anatomy of the uterus, it really lends itself to the concept of a lavage, the ability to wash the inside of the uterus to collect cells and potentially cell-free DNA that a tumor may be essentially emitting or part of that mix. Now, again, you're going to catch a lot of normal cells, but if you think back to 70 years ago, this was really the concept of a pap smear. So pap smears essentially just touch the surface of the cervix and then look for the presence of cancerous cells. The concept had been tried for many years and many different iterations for doing this uterine lavage, But it just never really seemed to work. Up until about a year or two ago, there were two really nice studies by other groups where they could show that they could collect some cancer-relevant material from a lavage. But our study was a little bit different. Our study said, can we do a lavage, but in a prospective manner? We don't know what the patient's diagnosis is, but can we look to find cancer in a patient who has not been diagnosed with cancer? So putting together everything we've spoken about to this point, here's a little bit of a cartoon kind of view. So the concept of the study is collecting material from a lavage where we would collect normal cellular material, normal cfDNA, but also cancer cells, potentially cancer cell-free DNA from this lavage. We would process it, we would isolate all the DNA, we would use a panel-based approach where we could get really ultra-high coverage, 5,000x coverage and above, look for mutations in cancer driver genes, and then compare the results of the DNC, the gold standard, versus our molecular analysis. And so this is the study. So the flow chart showed we enrolled the first 107 patients who were coming in for a diagnostic hysteroscopy. In other words, the physician had sent these women to have a diagnostic hysteroscopy. The suspicion was high for the presence of cancer, but it was unconfirmed. And then we would split the women, we would take the fluid and the collection material in a split manner, in the sense that the lavage fluid, which was being generated through the hysteroscopy, at the point when the hysteroscope is actually placed into the uterus, saline is used to open and distend the uterus, so the surgeon actually has a view inside. That material, that saline fluid, is normally just considered waste. We collected that material, centrifuged it, put it into two separate, essentially, bins, one that had cfDNA, essentially an acellular fraction, and a cellular fraction. And then we collected the DNA from each of those, and we did our high coverage sequencing using an endometrial cancer panel. In the meantime, and just following along with standard protocol, a DNC was performed on the women, that material went to pathology, a diagnosis was made. At the end of the study, we then compared the molecular and the histopathologic results, and this is what we found. The women that were coming in were of a normal distribution one would expect to be examined for presence of endometrial cancer. You can see the age distribution, the race ethnicity distribution, the BMI, in other words, whether these women were considered overweight, obese, thin, because again, there's a very strong correlation with body mass index. The higher the body mass index, the greater the likelihood of having endometrial cancer, and the menopausal status. About half of the women were premenopausal, about half were postmenopausal, slightly more postmenopausal as you would expect in this population. The preoperative diagnoses, in other words, going into the OR, the surgeons had written in their notes that about 50% of the women had abnormal bleeding, including postmenopausal bleeding. About 30% had uterine polyps, so an actual structural variation, and then you can read what some of the other preoperative diagnoses were. This is the panel we started with. So our colleagues at Swift Biosciences have a 56-gene cancer panel. Now, they have the amplicons that either cover the entire gene, for example, P53, or hotspots within a number of cancer genes. So we could use this panel, and indeed we did use this panel for some of the first patients just to see how much DNA we were getting, the quality of the DNA. But again, with Boris Riva's help, we were able to actually use this panel and even constrain it further, such that Boris came up with a 12-gene panel. And you can see I've highlighted the genes with red boxes, and we actually added two other genes, ARID1A and PIK3R1, to the panel. There were 102 amplicons, an average length of about 138 base pairs. So really, it's a very small panel that we used for this study. This is essentially the final tissue-based histopathology diagnoses of the 102 women from the 107, 102 that met all criteria. This was the final diagnosis. What I would point out to you, there are 7 women who were diagnosed with endometrial cancer from the 102 that finally were enrolled in the study. 6 of those women had stage 1A endometrial cancer. That's early microscopic cancer. The grade of the cancer, 4 of the women had grade 1. Again, very early, very low grade cancer. And you can see what some of the preoperative diagnoses were, the age of the women, and again, the BMI of these women. This is actually what some of the tumors look like from the D&C. When we went back to the pathologist, this is Tamara Kalir and her colleagues, we could actually look at some of the specimens. And what I would point out for you, and particularly in D, where you see that I've circled that, that's actually the tip of a polyp. And within that polyp is the cancer. So actually even hidden within this structure, there's a cancer. Now, when we went back to the molecular analysis, this was very exciting. All 7 of the women that had a histopathologic diagnosis of cancer, all 7 were found to have mutations. I've highlighted 3 of the patients here, patient 396, 451, 468, I've color-coded them. These are some of the genes, the actual mutations, the mutation class, whether they were drivers or potential drivers. And then I've got 2 columns here, cell pellet DNA and cfDNA, just to show you the coverage that we're getting and the allele frequency range. So the range goes from about 1%, which was our cutoff range, yes. So that really is the bottom of, there were things that were lower, but we used the cutoff of 1%, even though we had a very high coverage amount, all the way up to about 40%, 38.8% here. So the range, the allele frequency range is really quite broad. And as you can see in some of the patients, it's actually quite high. So it really made up a large percentage of the DNA that we were identifying. So the range was quite high. The other thing I wanted to highlight too is that, again, remembering that all 7 women with pathology-confirmed cancer, all 7 had mutations. And when you look at the cell pellet DNA and the cell-free DNA, in almost every instance, if you found mutations in the cell pellet, you'd find them in the cell-free. But that wasn't always true. In a couple of instances, and I've highlighted these by arrows, you may have found mutations in the cell pellet, but not in the CF. Or conversely, you may have found them in the CF, but not in the cell pellet. And again, I think the importance of us having done these 2 fractions is highlighted by this kind of an analysis. Now, the lavage fluid mutations that we had identified, we were able to go back to only 4 of the actual specimens because there wasn't enough material for pathology to allow us to extract anything for research purposes. But the majority of mutations that we identified in either the cell pellet or the cell-free DNA were present in the tumor. And you can see that by the some of the mutations I've highlighted in red. They're present in pellet, they were present in the CF, they were present in the tumor. But again, in some instances, for example, I've highlighted with the green arrow, there were mutations in the tumor which were not detected in either of the cell-free or the cell pellet. Sometimes you would find things in the pellet, not the tumor, or in the cfDNA in the tumor. And again, this probably, that's not proven, probably has to do with the amount of mutation that we're seeing, and ultimately that has to do probably with the heterogeneity of the mutations present in the tumor. Again, not proven, but that would be our thought. Now, I think that speaks quite highly to the potential of using this as a screening tool or as a diagnostic tool. In other words, 7 women with cancer, all 7 had mutations. But really then the question becomes, what of the other women in the study? So I've highlighted in this table the final histopathological diagnosis of the women. So you can see again 7 women with endometrial cancer, but the majority of women were diagnosed as having polyps or polypoid fragments, normal endometrium, fibroids, polyps and fibroids, endometrial hyperplasia, and other. So what did their mutation characteristics look like? This was really the surprise of the study. Half of all women, or 51 out of 102 in our study population, had significant driver mutations in their uterine lavage samples. So you can see I've tried to highlight on the upper bar the women that had cancer, the women that had endometrial hyperplasia, polyps, myomas, normal that had significant cancer driver mutations. And you can again see that some of these mutations are in KRAS, P10, PIK3CA, P53, ARID1A, RB1. So these mutations were prevalent, and they actually were there in particularly high allele fractions. Now, again, because this is new technology, and again, we're using next generation sequencing, one thing that was very important for the study was to validate what we had done by an orthogonal methodology. We went back and we pulled about, I'd say, about a third, a little more than a third, of all the variants that we'd identified. If the allele fraction was greater than 10%, we confirmed in the original sample by Sanger sequencing. If it was less than 10%, we did ddPCR. Every mutation that had been identified by next generation sequencing was confirmed by either Sanger sequencing or droplet digital PCR. In other words, the mutations were real. They were not artifacts. You can see by the chart here the allele frequency. What I'd also like to point out is that when we went back, we could actually identify some new mutations by digital PCR, which obviously can go much lower in, it's much more sensitive. We can actually use that, and I've highlighted some of those cases in blue, where again, we can identify mutations that we had missed by NGS in either the cell pellet or cfDNA. Now, what did the frequencies look like in these women that did not have evidence of cancer by pathology or histopathology, but we were finding mutations? And well, the allele frequency range really ranged from 1 to 30%, which an average of about 3%. And you can see on this histogram, the women that did have cancer, we've highlighted those women with these orangey colored boxes. All the women that did not have cancer, we've highlighted just essentially by just typing their numbers here. So you can see the majority obviously did not have cancer or didn't have a diagnosis of cancer. But the mutation spectrum, whether they were driver mutations, potential drivers, mutations of unknown significance, were really quite striking. And indeed, I've just highlighted two representative cases. So here's patient 395, who had mutations in a total of 5 genes, 12 driver mutations. The genes affected were P10, PIK3R1, PIK3CA, KRAS, and FGFR2. And again, the allele frequencies were really quite, pronounced, 1 to 7%. Patient 485, who had a benign polyp, 4 driver mutations, and affected a total of 2 genes, KRAS and PIK3CA, and again, the allele fraction was 1 to 3%. So looking at it, it's very difficult to understand the pattern of why a benign polyp or benign polyploid fragments would have these significant mutations in cancer driver genes. Now, does that mean that every woman that has a mutation will not develop cancer or doesn't have cancer? Or does it mean that every woman who has a mutation will eventually develop cancer? Well, in a group of 100 women that have undergone hysteroscopy, you would expect about 10% to have endometrial cancer. Maybe 1 or 2 more percent will develop endometrial cancer. But 50% of random population of women undergoing a diagnostic hysteroscopy should not develop endometrial cancer. But what's interesting, we did have one patient who returned about 10 months later, who originally had her surgery in 2000, hysteroscopy, so we call it surgery, in 2015. She was diagnosed with thickened endometrium. We had identified 5 driver mutations in 4 genes, but pathology didn't see any evidence of cancer. Patient 10 months later returned for postmenopausal bleeding. Pathology revealed this complex hyperplasia pattern with the tibia, which is suspicious for low-grade endometrial adenocarcinoma. And at 11 months after that initial lavage with that molecular analysis, of grade 1, stage 1A endometrial cancer was removed. And there was no myometrial invasion, but there was a cancer. And what I've shown you in the inset is a picture of that cancer. So there is potentially some predictive value in this. The problem is the noise, we'll call it in the background, is was too high. And if I were writing this, I would put quotation marks around the word noise. We don't know what that noise is. Now, the only associations we could find with the presence of mutations were really age and menopausal status, and clearly the two are linked. But the older you were, and if you were postmenopausal, you were more likely to have these cancer driver mutations. Now, to give you a little bit of perspective on the problem and the power, this is actually one of the patients. So here is what the DNC and then what the pathologist looks at from the slide. So these slides are all from one patient. If you look at the second row, if you look at slide number 17 and 16 there, if you've got good eyes and you've got a good video monitor, you'll see there's actually some little black dots left by a felt tip pen. That's the patient's cancer. Those areas are where we were able, well, we can't say the areas, but we believe those cancers were the ones that actually, in essence, emitted the DNA or the cells that we were able to detect by the molecular analysis. So the power of the approach is really quite high. The sensitivity to detect these mutations is very high. In conclusion, and really trying to think about future work, let me just highlight three things. So I think the study provides the first evidence for the ability of an NGS-based approach to prospectively detect early stage endometrial cancer. And again, not to just keep hitting this over the head, but the point is these were microscopic cancers detected on essentially saline fluid that was considered waste material. We could use a panel which was designed specifically for endometrial cancer to detect these mutations. The study also identified a previously unknown but highly prevalent landscape of driver and potential driver mutations in women without histopathologic evidence of endometrial cancer. Again, remember, 51 women had mutations in cancer driver genes. What does this mean? I mean, when you think about it, this complex genetic environment, what does it reveal about our current understanding of detection of genetic mutations in tumorigenesis, predicting the clinical consequences of mutation detection, and how will we make patient care decisions in light of these findings? It's not enough just to say, yes, there's a mutation, yes, we can validate it, we know what we found is true. How does that play into the reality of cancer development in these women? So just on these last slides, I just want to leave you kind of with a thought, and really this is, our understanding of the development of cancer. Clearly, and this is a cartoon that I'd taken from a Nature Reviews Cancer article. So from the left side of the panel, normal tissue, to the right, invasive cancer, and in a number of different tissues, there are a number of different, pathology or histopathology, recognized features that we can, go through the entire length of spectrum from normal to cancerous tissue, and really some of the findings and some of the timelines that are there. Now, that really plays into our understanding of the development of cancer. But what I would ask you to think about is, if we think about the endpoint cancer, and so what I have is this cartoon of, let's say cancer standing in the doorway, we see the shadow of cancer, and if we then take that cancer material, as we've done for so many tumors now, and as we always do for, understanding cancer development, we take those tumors, we sequence them, and we can bring all, the different technologies from single cell understanding to next generation sequencing to really break down those tumors and understand the genetics of it. What are the features that are there in that last time point when we have a cancer? And so we have to be careful not to look back down that tunnel and say, yes, here we have these mutations in the tumor. So if I walk backwards, whenever I see one of these mutations, I know I'm on this path towards the development of cancer. It's irrevocable. When I see these mutations, I'm leading here. But that's really not the way we should be thinking. I would say, from based on our findings in endometrial cancer. Just having the mutations and seeing them in a timeline does not mean, therefore, that you will develop cancer, because, again, we're finding these mutations in women who do not have evidence of cancer. So it's not a fair model to say cancers have these mutations. If I look backwards, any time point along here means if you have these mutations, you will develop these cancers. It's an easy trap to fall into. And again, I think the study really highlights we have unparalleled technology now. Next generation sequencing is leading to a new revolution in understanding. Our study was in endometrial cancer, but if you go through the literature, you actually see very similar studies for leukemias and skin cancer. So again, I wanted to thank all the patients and their families that participated in our research program, in particular the people in my laboratory from the endometrial group, Navia Nair, Olga Camacho Venegas, in particular Peter Dettino from the Division of Gynecologic Oncology, the people at Swift Biosciences, in particular also from the ICANN Institute for Genomics and Multiscale Biology, Boris Riva, Bobby Sebra, Dmitry Rykhanov, a number of people that helped fund these studies. And even for a study like this with only 102 patients, really all the attendings, the fellows, the residents that really helped this study come to fruition. So with that, I wanted to thank you for your patience and open it up for questions if there are any.
Thank you, Dr. Martignetti, for that informative presentation. It's time for Q&A. If you have a question you'd like to ask Dr. Martignetti, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question in the box that appears on your screen, and click on the send button. We'll answer as many questions as we have time for. If we are unable to get to your question, Dr. Martignetti will follow up with you via email after the presentation. Let's get started. Our first question is this technology available for screening women now?
So thanks, Julie. So the technology is currently available, but it is not ready for prime time. It's really not ready to screen women because I think, again, if we encapsulate the study findings, we can detect early stage cancers, but we've detected this landscape of mutations that we don't understand the biology and we don't understand the clinical relevance yet. So like in any good screening tool, what you'd really like to be able to do is act on the findings and the output. And we're at a point where we would be identifying women who do not have cancer, probably will not develop cancer, and we wouldn't know what to tell them. Really what we want to get to is the point where we can understand what these mutations in women without cancer really means.
Thank you. The next question is, what does having a cancer gene mutation mean for the patient?
Well, yeah, that's a great question. And quite frankly, we just don't know yet. What we're finding is that as we look in this population of patients, in other words, women, where we're looking at endometrial tissue, the results parallel what others are seeing looking at skin and P53 mutations, or looking at blood and looking at known leukemia cancer drivers. These mutations seem to accumulate, but they don't seem to lead to cancer. So we don't know what it means yet clinically. Really, that really becomes kind of the point of the a larger study, collaborative study, looking at larger numbers of women and following these women over a period of time.
The next question is Is this the only tissue that has been found to have mutations in cancer genes without pathologic evidence of cancer?
Well, thanks, Julie. So, I guess kind of an answer to the previous question, it's actually not. As I mentioned in the answer to the previous question, there are other tissues. For example, in skin, when skin has been removed, let's say from the upper eyelid, people are undergoing plastic surgery. You can actually look at that skin and go really deep again, and break up a small piece of tissue into small areas. And you can sequence for the presence of p53 mutations. And again, you find entire clonal patches of p53 mutations. But again, when you look at the skin, other than just aged and sunbeaten, there's really no evidence of any cancer present. It looks like normal skin. Same with blood. In some of the large number of studies, I think about 4 different studies, looking at a total of more than 20,000 patients where people do GWAS studies for different psychiatric disorders. Kind of as an unexpected outcome there, people had identified a small percentage, but still a relatively strong percentage, 3-4% of individuals that carried mutations that were known leukemic driver genes, cancer driver genes. And the individuals that submitted these samples had no evidence of cancer. Now, that's 2 examples. It's actually even been identified in a very recent study that women that were undergoing a lavage of their peritoneum that were for ovarian cancer. So there were 2 groups of women, a group with ovarian cancer that were BRCA positive, a group that did not have ovarian cancer. When you actually looked at the peritoneum of these women that didn't have cancer, again, you found p53 mutations there, a very low fraction, but that was probably diluted given the amount of surface area that's there. But again, you found p53 mutations there. So my impression is that the more we look and the more we bring these technologies to bear and the more we start using very high coverage of particular areas of genes, the more mutations we're going to find. I hope that answers the question.
Great. Thank you for that answer. We'll wrap up with one more question, and that question is, what next?
What next? Well, again, is part of the other questions and the other answers I've given. Really, I think it takes a couple of different forms. I think one form for what next is really to follow the women in the study that we've already identified these mutations over time. What becomes of these women? What percentage will develop cancer, if any? What other health issues may these women have, if any at all? So that would be one part of the study for the future. I think the other study is really to do a larger study. More women, different gene panel. Given what we've identified in the 12 gene panel, I think it makes sense to potentially open up the gene panel to look for other kinds of genes that may be involved in proliferation of cells, immunology, immune response, a number of different things that we can interrogate for from women. And it would probably make also sense to look at the timeline of women who do not have any evidence or any suspicion of cancer. What does the normal endometrium look like? What do the different areas of tissue teach us about the accumulation of mutations, if any mutations are truly accumulating? I think, again, what this study does is really point out the development of mutations in cancer genes. In what we had seen as small panels, we can imagine that in a larger panel there'd be more mutations, but it really identifies the accumulation of mutations, not just in this tunnel approach that we think about for cancer, but really maybe as part of normal development. And what are the forces, if any, either from within the cell that prevent cancer from arising, despite the presence of these mutations, or forces externally, again, immune system, whatever it may be, the environment, that could be suppressing these mutations. I think that I think it would be a quite exciting period to see, what this kind of opening of a window in this identification of mutations will really teach us long run about the biology of normal tissue and potential transformation, carcinogenic transformation.
I would like to once again thank Dr. Martignett for his presentation. Do you have any final comments?
Well, first of all, I wanted to acknowledge the format of this. And I think having traveled a lot, I think the format is quite nice in the sense that people can just log in and hopefully be exposed to something they may not be able to travel to hear about, and the same to be able for me to get questions from different individuals. But I really do think what I'd like to leave as a take-home is we're really at an exciting time where NGS and panels and really a lot of the new technology is really going to open a new window for us for understanding, probably the way maybe 100 years ago when people first had access to x-rays and we could look inside bodies, and the level of understanding jumped incredibly. But it also raised a lot of questions. Well, what is that? Is that normal? Is that not normal? As we got to CTs and PET scans and MRIs, more recently, again, it opened up a whole new knowledge base for not only diagnosing, illness, but really maybe understanding normal development. And again, I think we're really on that verge of understanding again, with NGS and really the application, the availability to do these kinds of studies, we're going to be opening up a lot of doors. It means a lot more questions and a lot more kind of, head scratching and really periods of thoughtfulness. But again, I think this is really the exciting part. We're on the verge of really being able to break in, break down, and really understand things that we've never been able to see before on the genetic side of tissue growth and development.
Thank you once again, Dr. Martignett. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through July 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.